We are so grateful that Dr. Wilhelm would make time in his schedule. He's been busy with NSF all week, and he broke away from a Zoom conference in order to be with us. And as soon as he's done, that is after 1 o'clock, he's going to go back and resume his conferencing. This is a very busy because a very distinguished man, as you will quickly hear. Got his undergraduate degree in genetics from University of Western Ontario. Went on there at the University of Western Ontario for his PhD in plant sciences. He did a postdoctoral fellowship in marine sciences at the University of Texas, Austin, another postdoctoral fellowship at the University of British Columbia. He came to the University of Tennessee in now let me make sure I've got this right, 1998, is that correct? 1998, and has been also graduate director and associate head of that department. He has won the Science Alliance Faculty Award for Research here at the University of Tennessee, also the Chancellor's Award for Research and Creative Achievement. He has been responsible for more than $20 million of grants coming to the University of Tennessee, mostly from the National Science Foundation. He holds the Mossman Professor of Microbiology, distinguished professorship. And we're so grateful to have Dr. Stephen Wilhelm with us today, speaking on the threat of toxic algae blooms in our fresh waters. Thank you.
Thank you, Professor Littman. As always, and once again, it's hard to follow on your introductions. They're very flattering, and I appreciate it. I'm going to hold that to one day that I am going to hire you to follow me around and enter rooms just before I walk in, and to say these wonderful things. But today, what I want to tell you about is not necessarily about me, but about the great work that graduate students, postdocs, research associates, undergrads have done in my lab over the last 23 and counting years at the University of Tennessee. I'm going to talk specifically today about cyanobacteria in freshwater systems. I was lucky enough to be here 2 years ago, and we had a wonderful conversation about giant viruses in the world's oceans. And today we're going to talk about this different topic. Okay, oh, gotta use a different button. There we go. So what are cyanobacterial harmful algal blooms? Well, what I want to talk to you about today is events that occur when we have an overgrowth of organisms in freshwater systems. You can see some pictures here. These are from Lake Tai or Taihu in China, one of our study sites, and I'll introduce that in a minute. But these systems, we have them in North America, we have them all over the world, and I'll show you that in a second, are responding to nutrient pollution that's driving an overproliferation of single-cell photosynthetic organisms. And they always say a picture is worth 1,000 words, so perhaps a video is maybe worth 1,000 pictures. And so what I'm showing you here is a small boat we work with in China. This is from October 2014, and you're looking at us transiting through a canal in the lake, and that green material in the water is not paint, that is growth of cyanobacteria in the system. The cyanobacterium here is called Microcystis, and it will grow to densities as high as 1 million or maybe even 10 million cells in a milliliter of water. As you can see, these blooms are massively disruptive. Along the back wall there, we see a number of fishing boats where people are fishing for commercial sale, but also for subsistence. So these blooms can cause a great problem. If we take a closer look at these blooms, what we find out is that it's really a mixture of different algae growing in there. Now, typically speaking, the blooms are dominated by one organism. You can see Microcystis over here. I'll show you a bit more of that in a second. But we always have to keep in mind that there are other organisms growing in the system. And one of our jobs is to tease apart why we get certain organisms versus others. The one I'm going to spend most of my time talking about today, and the one we focus on in the lab mostly, is Microcystis. Now, Microcystis is a globally distributed cyanobacterium. It has a number of different species. It's been reoccurring as a problem in the Laurentian Great Lakes since 1995, and it produces a compound, a secondary metabolite that is hepatotoxic, so it attacks the liver. This compound was first called fast death factor. We don't call it microcystin, but I love the impact of its original name. Beyond microcystin, the organisms produce other secondary metabolites. One we were able to show a number of years ago in work by Emily Rogers has endocrine disrupting effects. So think birth control pills and water. It actually causes larval zebrafish to prematurely sexually differentiate. And even without these compounds being produced, as you saw, these blooms are large. They're massive in scale, and they can disrupt food webs and change water column physiochemistry. In short, they're very undesirable. This is not just a problem in China or just a problem in a couple of small regions. Over 108 countries around the world have had confirmed bloom events for Microcystis. And in fact, of these 108 countries, positive toxin tests have occurred in about 80. So this is a global problem, and we think a growing global problem because of climate change. And I'll address that in a little bit. If we zoom into the map of the United States, we can see that there are reports of toxins all over the United States. Now, one thing that jumps out at me is we're right here in Tennessee, where it says there are no reports of cyanobacterial poisonings based on the USGS data. However, I would point to two studies that have clearly shown the presence of toxic cyanobacteria in Tennessee. A study by Wayne Carmichael, one of the sort of leaders in this area, up to his retirement a couple of years ago, found that in the Guntersville Reservoir, which is on the Tennessee-Alabama border, there were filamentous cyanobacteria, in this case, Ligambia wallii, that were producing toxins. Perhaps more salient to us in Knoxville, Tennessee, Andy Willett, a postdoc with me in the early 2000s, was driving by UT Hospital one day and noticed a big green scum on that retaining pond out front. He took a sample, and that sample ended up getting worked into his workflow, and he was able to demonstrate that there were toxic cyanobacteria here in Knoxville, Tennessee. That study is somewhat salient to us, is that because it was really when Andy was developing the molecular tools we started to use in the lab 17 years ago. And it's not just a spatial consideration here, but there's a long history of these freshwater blooms. There are stories from China of a general who parked his army on the side of a river, and the entire army fell not by the enemy, but it was taken out by the green water that they drank. There are recordings of this in art, Dutch masters having painted in the 1600s bright green lakes, excuse me, where, in some cases you would see they had blue paint. That was always a question. Maybe they didn't have blue paint. They did have blue paint because you can see it in the skies. The two systems I'm going to talk about today, one is Lake Tai in China, which has been reporting reoccurring blooms since about 1960. And then I'm going to talk about Lake Erie a little bit. We've been studying it since the late 1990s, and they had problems in Lake Erie that culminated in a water supply shutoff. So where are our study sites that we focus on? Well, one thing that Mark didn't talk about, and he always does take a jab at me, is I'm a Canadian. My ancestral family home was right about where the red dot is, somewhere in here. So I grew up just north of Lake Erie, and we've studied Lake Erie in the lab since I've been at Tennessee. Lake Erie is the 12th largest lake in the world. Its drainage basin is home to about 11.5 million people. And there's a lot of idiosyncrasies about the lake. It's the smallest, it's the shallowest, it's the warmest. Not listed here is it's actually also the coldest. It's the most productive, and it's the most human impacted. One interesting thing about the Great Lakes region is if you took all the states and the two provinces that touch the Great Lakes on any shoreline and made them their own country, they would be the 8th largest economy in the world. And they all depend on the Great Lakes for transportation, for resources, et cetera, and so forth. So it was quite a bit of a problem then in 2014 when water was coming into the city of Toledo, Ohio, which is about 400,000 people. They detected toxins from cyanobacteria in the water supply. This caused a shutdown of the water supply to the city of Toledo during a long weekend in August of 2014, when people were expecting to go out, enjoy the beach, enjoy being with family and friends on a holiday weekend. Instead, they were completely without water. This forced businesses in the city to shut down. It forced industries that were making foodstuffs to shut down. One thing that shocks a lot of people is that hundreds of thousands of liters of beer were thrown away because they were potentially made with toxic water. So this was a big issue. Another place we do a lot of work is in Taihu, or Lake Tai in China. It's shown right here. The drainage basin of Taihu has about 40 million people in it, includes the large cities of Wuxi and Suzhou, each that are about 9 million people right now. Also, of course, Shanghai, one of the fastest growing cities in the world, is right here. This lake, as I mentioned earlier, has had problems since the 1960s. And in fact, the late 1990s, they had to shut down the water supply to Wuxi for multiple weeks because of toxic cyanobacterial blooms in the system. So for these reasons and many others, we've been focusing on toxic cyanobacteria, asking questions about why we get these blooms and why the cells are even making the toxin. So I want to go through this, and I want to kind of break up the talk into things that we know for sure and things we think we know. And I think that's really important. Science has been under fire a little bit lately in terms of what scientists will say or won't say. And for the most part, it's because we're trained to never be 100% sure of things if there's any question at all. We just won't say those things. But there are some things we can say for sure about cyanobacterial harmful algal blooms. They are ancient. We think cyanobacteria were the first photosynthetic organisms on the planet. There's evidence that goes back a couple billion years on this. People do not like them. These bloom events, as I've illustrated, they disrupt water supply, they disrupt people's vacations, they're hard on regional socioeconomics. Blooms are tied to multiple environmental conditions. A quick survey of the literature clearly shows that changes in nutrient flow, changes in the environment are all having effects on these blooms. Humans are having an effect, and I'll come back to that one in just a second. And I think the one thing we're most confident of is that we have a long way to go before we actually can control these bloom events. So a common question I get is, why do you say there are human effects? Well, one study that Johanna Rinta-Kanta, one of the first PhD students to work on this project did back in the early 2000s, was to look at Microcystis cells in sediments of Lake Erie. She was able to go back almost 100 years and look at the types of cells that were there using molecular tools. Now, the current blooms in Lake Erie have started in 1995. But in going back almost 100 years, Johanna was able to show that the blooms that are present now, those same cell types were there in the early 1900s. So they're present in the early 1900s, but they weren't blooming until the late 1990s. Well, we do know that Lake Erie is one of the most managed systems in the world. We work to control the water levels, we work to control transport on the surface, we work to control everything about the lake. So something we're doing, on purpose or not, is influencing the communities that are arising in the lake. There's an old adage in microbial ecology that says, everything is everywhere, but the environment selects. What this suggests is that microbes can get to all parts of the planet, and that what is happening in the local environment determines which organisms can proliferate. In the current case, we know that Microcystis has been in this system for a very long time. And whatever's been happening since the late 1990s is resulting in Microcystis blooms. And in fact, at the end of her paper, Johanna included this fairly salient line, everything is everywhere, but human activity selects. And so we are quite confident that it's some sort of human activity, be it associated with nutrients, be it associated with climate change, and many other possibilities that's causing these blooms. I think what is more salient and people are more interested in what we think we know. What are the directions our science is going? What are the questions we're trying to ask to try to deal with these bloom events? And there are a number of things. We're fairly confident that nutrient loads, runoff from agriculture, from urban centers, from golf courses, changes in water flow patterns that result in nutrient loads are causing bloom events. We know that cyanobacteria in the laboratory do much better under warmer temperatures that are consistent with our projected climate change scenarios. We know that the time of season clearly influences bloom events. We also know that things like pH influence bloom events. In fact, these Microcystis blooms often push the pH of the water up over 9.2. And there's some positive feedback in that, and I'll mention that in a little bit. We know that meteorology, rain, wind, storm events all influence these blooms. And we know that agricultural chemicals, farms, golf courses, urban green space, all influence bloom events. What this leads us to is this mantra we've developed in the lab that these blooms are complicated. We can't just pick on one thing. And so we're slowly trying to work through and understand how each of these types of parameters might shape the blooms that we're seeing. So one of the things about my job I do is I go to a lot of meetings. I'm in a lot of conference calls, as Mark pointed to. And one of the questions you'll get, especially from people in the media, is why can't scientists simply agree on what's going on here? And I think one of the issues I wanted to mention is that scientists use different tools, and these tools work across different scales. In my lab, we may use tools that are involved with DNA or RNA sequencing or culture work. Others use satellite imagery to predict what's going on and try to understand these bloom events. And there's a lot of disconnects between what's going on at the level of an individual cell versus what's going on at the level of a large lake. And so it's really important for us to try to understand then how we can bring these ideas together. What other confounding things might we find in our experiments? Well, one thing I wanted to point out is that these blooms are incredibly patchy. What you're looking at here is surface waters in Lake Tai, and you can see that there's areas where the bloom is very thick and other areas where the bloom is non-existent. This creates a scientific conundrum. Do I sample here or do I sample here? Most researchers, maybe good or bad, will always try to go where they see the bloom event because that's what they're there to study. But what's going on here is just as relevant as what's going on over here. And it's not just at the surface. One of the challenges of Microcystis is that they can migrate vertically. What you're looking at here is data looking at depth in the water column across biological time. And what you can see is a bloom of Microcystis moving up and down in the water column and migrating. They do this by having gas vesicles that make them buoyant and then ballasting themselves by photosynthesizing a lot and producing a lot of photosynthates. And this allows cells to move up and down in the water column and make fine adjustments in the light field they're experiencing. Remember, these are phototrophs, as well as making fine adjustments in the nutrients they're consuming. Maybe they've consumed all the nutrients at the surface, and now they want to consume the nutrients deeper down. So all of this is complicated by the fact that there are all sorts of changes going on in the Great Lakes Basin. And I think it's important to think about those changes in respect to what's going on with Microcystis blooms. These changes, as I've noted, have only been going on for the past 25 years. The first one everyone jumps towards is the fact that the Great Lakes are warming. This image is blown up to show 1995 to 2018, but you can go back even farther. The average temperature of the Great Lakes is slowly creeping up. This is really important because cyanobacteria do better under warmer conditions, although I'll talk about what cold events can mean in a bit. The other thing that this warming means is that the weather is not the same. It means water is not always now where it has been in the past. This leads to a lot of changes in the regional weather patterns and climate. We think that going forward, climate change is going to give us a lot more in terms of episodic storms that are going to disrupt the system. These storms can create massive changes in surface water temperatures. They can introduce runoff from farmers' fields and areas outside the lake. They can even resuspend sediments, which are also chock full of nutrients that make algae grow. Now, all these nutrients don't come just from nature. One of the things that's happened over the past few decades is that we've really become dependent on the use of fertilizers for agriculture. From this National Geographic article, we can see that nearly half the people on the planet probably wouldn't be here if we hadn't been putting fertilizers in soils. More shocking and more probably important to us is that we've changed the types of fertilizers we use over the past 25 years. In the early 1900s to the mid 1900s, ammonium nitrate was the common fertilizer of use. Then in the early 1980s, 1990s, we really had a big switch over to urea. Urea is easier to handle, it's more cost effective, and it's less dangerous to use than ammonium nitrate. Interestingly, the switch from ammonium nitrate to urea corresponds with the proliferation of Microcystis around the world. And so that's been something we've been very interested in, and I'll get back to that later as well. Other things that have changed are farming practices. We've moved away from the standard mom and pop farming practice to more large-scale industrial farming practices around the country. This is especially true in the US and in Canada. What this means is that some historical practices, like using winter wheat to cover fields, has dropped immensely. What you're looking at here is a figure from a paper we published with our friends in Ohio, George Bullerjohn and colleagues in 2016, looking at acreage of winter wheat from 1970 through to 2015 for the states of Ohio, Indiana, and Michigan. In each case, you can see a decline in the winter crops. Generally speaking, most people think that these winter crops are important for retaining nutrients in the soil and preventing those nutrients from running off into lakes. One of the other changes that I'm not going to talk about today is that in these same regions, the use of drainage tiles in fields has massively exploded. And we see way more farmers' fields with tiles now, creating sort of highways for nutrients and water to run off fields and immediately into lake systems. Okay, so let's get into the science that we're interested in after that little bit of background. One thing we think about in the lab is that what we're really interested in is trying to figure out what the cells are doing. So instead of us doing a bunch of silly experiments that may not tell us what the cells are doing, we try to ask the cells themselves what we're doing. We do this by using the techniques of modern molecular biology. The molecular toolbox for cyanobacteria keeps evolving, but we now have the tools of genomics, transcriptomics, proteomics, metabolomics, all of these things that tell us about the potential for organisms, the regulation and the function that organisms are trying to undertake out in aquatic systems. So how do we use these tools? Well, you're looking at a figure here, a mock-up made by Xiaoming Tong, a colleague of ours from China who spent a year in the lab looking at taihu. In 2014, we worked with our Chinese colleagues to carry out a spatial and temporal survey of the large Microcystis blooms they had, and in the process, we generated 1 billion RNA sequences. We analyzed those sequences and we realized that there were bloom events going on that could be distinguished between bloom development, so when the cells were growing very fast, and then bloom maintenance, when the cells had taken over the system, but they were being forced to survive. What we can see here is that there's differences in regulation that occur in the system, and this starts to give us some ideas as to how not only Microcystis can take off and grow quickly, but then maintain itself through several months in the summer. Now, when I talk about Microcystis, I should point out that this just isn't one cell type. And work from Lena Pound in the lab has shown this. Often we talk about these as an individual or a couple individuals out there, but Lena has clearly shown when we've looked at taihu, all of these bars here, we're looking at a phylogenetic tree on this side, are individual Microcystis types that she's found in taihu, and she's been able to tie them to different times of the year. You're looking at samples here from June, July, August, September. And then she's also been able to enumerate these based on the transcript copy number in the samples. And what we begin to see is that there's seasonal sweeps of one type occurring early in the lake, and then other types moving in later in the year. This information is really important because not only have we been able to show now that the organisms are trying to do different things or are doing different things at different times of the year, that it may even be different subpopulations that we don't recognize by just looking at them in a microscope in the system. And doing different things is something that Microcystis has been become very well known for. One of our serendipitous observations in the lab was made by Sunny Peng and Robbie Martin, where in storing some samples in the lab on a bench that became cooler in the winter months, they noticed the cells became more toxic. They followed this up and they've been able to show that as you drop temperature from Microcystis from 26 degrees to 19 degrees, that the cells become more toxic. Robbie's followed up on this recently and been able to show that when you drop the temperature, the cells almost immediately start upregulating a gene that's responsible for toxin production. Here it's the MCYD gene, and that about 48 hours later, microcystin production increases as well. This tells us a couple things. One thing it tells us is that there are some predictive values in the data that we're generating, molecular biological data, but that there's a temporal offset. You cannot do direct correlations between the transcripts that are present and the amount of a compound in the cell. What's really exciting about this is that this delayed stress creates a predictive value, that when researchers start collecting this type of data on a regular basis, they will be able to predict, perhaps with 48 or 72 hours advance notice, that a toxic event is about to occur. Does this actually work, or does this really work? Well, for the paper they published a couple years ago, Robbie built a really crude model where he modeled surface temperature in the lake, the measured microcystin in the lake, and the predicted microcystin in the lake. And I want to say this is a really, really crude model that was built, but we can see that this temperature decline, potentially tied to a storm event, coincided with a spike in toxin in the system. Now, over the last year, Robbie's been working with me and colleagues in Germany, where we're trying to put this type of information into an agent-based model, so a model that looks at individual cells to determine when they make toxin and when they don't make toxin. The challenge now is to incorporate this into existing models that predict the presence or absence of the cells. So not only can they predict presence or absence, they can predict whether or not the cells are actually going to potentially be making toxin. But as scientists, we're not just interested in this ability to predict. We want to understand, and we want to understand the mechanism. And so Gwen Stark has been working in the lab the last year. She joined us, I guess, last Christmas. And she's been working with Robbie on this same question. And she's been asking the question, what does cold temperature actually do? And we've kind of settled on this idea of excitation pressure to this point. The idea being that when temperatures drop and the same amount of light energy is coming into the system, that the cells are continuing to photosynthesize, but now they're generating too much energy. And this excess energy leads to oxidative stress. And so Gwen's been able to show that this actually happens in the cell here, looking at PCC 7806, an isolate in the lab, where the warm grown cells here are 26-degree grown cells, have low excitation pressure, and the cold grown cells here have higher excitation pressure. They have excess photosynthetic energy, generating oxidative radicals, probably leading to the need for microcystin. Now, I want to point out this does not work in every culture we have, and we're trying to understand that. So then the last little vignette I want to tell you is I want to make the point that sometimes it's not what you can do or what makes you thrive, it's what you can do to survive. And to make this point, I want to introduce you to diatoms. Diatoms are single-celled eukaryotic algae. They're phototrophs. They compete with Microcystis for space in the lake. They're one of the great photosynthesizers on the planet. The great thing about diatoms is they have these amazing little skeletons here. These are only a few microns in size, but you can see they're highly detailed and they're made of silica. So they have these silica skeletons that scientists call frustules, and they need these to grow. So as part of their growth, they bring in silica from the outside, build these structures. We can visualize this with a dye called PDMPO. I've written out the scientific name here. You are welcome to try to pronounce it. I always butcher it. PDMPO is a dye that intercalates into these frustules. So as the diatoms are growing and they're taking in silica and building frustules, little bits of this dye get put into the frustule, and you can actually see new diatom frustule, the skeletons of these algae, being made. So Brittany Zepernick in the lab has been studying this, and she's happened upon something that's fairly interesting. Normally we grow diatoms at pH 7.7. This is the pH we'd often see early in the year in lakes. In Lake Erie, it might be a little bit higher. As I mentioned earlier, Microcystis, when it blooms, pushes the pH up above 9, above 9.2 actually often. What Britt's been able to show in the lab and in the field is that as you increase pH, the diatoms lose their abilities to grow. They can't lay down frustules, or they can't at least maintain their frustules. This gives Microcystis a competitive advantage. Now, there's multiple other competitive advantages Microcystis has at these high pHs, but in this case, Microcystis can survive this high pH where diatoms not. As a microbial ecology lab, the ecologists in us are really excited about this ability of Microcystis to outcompete the other algae. So where does this leave all this? This leaves us with one big challenge, and this goes back to the idea I posed earlier about different scientists use different tools. We're still not quite sure what the appropriate scales are to study bloom events. We are working at scales that range from a teaspoon to the size of an entire lake. Which of these scales will tell us the most about the blooms? We're not really sure. And when we talk about scales, we can't just talk about spatial scales. We also have to talk about temporal scales. Typically speaking, we monitor blooms on a period of weeks to months, but processes in biology can occur in seconds and in fact in milliseconds. So trying to understand what the drivers are of bloom events is going to require some sort of integration of this. What becomes challenging then is integrating all this information, integrating across scales of seconds to weeks, and integrating across a teaspoon to an entire lake. So all of that said, I think I will stop there. There are a lot of people to thank. In this slide here, there's some faces from the past at the top. Andy Willett and Johanna Rank-DeCanto got much of our Microcystis work going about 20 years ago. Today, that's carried out by Robbie Martin, Lena Pound, Brittany Zepernick, Gwen Stark, and others in the lab. Thank you very much. I'd be happy to take any questions if there are any.